Major techniques used for genomes characterizations. There were some major techniques which were used to characterize the genomes or to sequence the genomes and to study the function of the genomes. Some of the most important techniques which were used to characterize the genomes are cloning, hybridization, PCR amplification, sequencing, and computational tools. So these were some of the most important techniques which were used to sequence the genomes. In addition to these techniques, there were some other protocols which were used to sequence or to characterize the genomes. Genome characterization, one of the major technique was cloning or DNA cloning. In case of cloning or DNA cloning, genomes, they were digested with the help of restriction enzymes. They were inserted into the vectors to produce the genomic libraries. The examples of genomic libraries can be BIO502 and the libraries of some of the genomes were also in the form of cosmid vectors. So, cosmid vectors were used to prepare the genomic libraries. Bacterial artificial chromosomes were used to prepare the genomic libraries. Yeast artificial chromosomes were used to prepare the genomic libraries for the sequencing of the genomes. Another technique which were used to sequence the genomes was hybridization. To arrange as the genomics, large genomes were cut with the help of restriction enzymes and they were inserted into the box or yaks. Then those box and yaks were arranged with the help of technique which is called as hybridization. So to arrange large contexts of genomes to produce the genetic map and physical map, the technique which were used to align the context is called as DNA hybridization. So here we can see that genome characterization techniques, hybridization this is the genome of a particular organism. To arrange large context, this genome was digested with the help of restriction enzymes. So restriction enzymes converted the genome into small pieces. So these small pieces were arranged into clone contexts. So to arrange these pieces in the form of clone contexts, DNA hybridization technique was used. Another technique which was used to characterize the genomes or to sequence the genomes is called as polymerase chain reaction. So this is a technique by which we can amplify the DNA. There are different subvariants of this technique. Another technique which was used to characterize the genome is called as DNA sequencing. So DNA sequencing is one of the important techniques used to characterize the genomes. This technique was actually used to sequence the DNA or to sequence the genomes and to study the structure and function of the genomes. This is the chromatogram that how DNA or the genomes, when they were sequenced, we get the sequencing results. So DNA sequencing, DNA are amplified and sequenced. When we sequence the DNA or when we sequence the genomes, we see the peaks, like this is the blue peak of cytosine, a red peak is of thymine, green peak is of adenine, and blue peak is of cytosine, and this is the black peak, this is of guanine. So the sequencing data appears in the form of techniques, in the form of peaks, and each nucleotide is labeled, and there is a specific color of the peak, and with the help of these peaks we can read the sequencing data. Another technique which was used to characterize the genomes or to align the sequencing data once the genomes were sequenced, that is called as computational tools, or in other words we can say it the bioinformatics tools. So bioinformatics tools or the computational tools were used to align the sequenced DNA to produce the physical map of the genomes. So if we conclude that how genomes were characterized or how the genomes were sequenced, different techniques including the most common techniques cloning, sequencing, DNA amplification, and computer software were used to characterize the genomes. Till now, hundreds of genomes, including the genomes of viruses, genomes of bacteria, and many genomes of the eukaryotes has been sequenced and characterized by using these most common techniques. These techniques, they are also called as the techniques of molecular biology or the techniques of molecular genetics.